So welcome everyone for today's class. Today will be the last day for our workshop. So I'm quite excited with all of you. So we will not waste much time and start with our things. Um, so let me just quit the unnecessary stuff. So I have already sent you everything in the morning so that you can have it with you uh, before we start. So you have all the lectures of today with you. So So in this lecture, I will deal with CIB techniques, karyotypes, radiogram that we have left previously, crossing over factors, cytological proofing, and mechanism of crossing over. So all these topics will be covered after our what we have done yesterday. So let's start. So crossing over is just first was used by Morgan and Kettle in 1912. The exchange of precisely homologous segments between known sister chromatids of homologous chromosome is called crossing over. It is responsible for a recombination between linked genes and takes place during the Pachytene stage of meiosis, after the homologous chromosomes have undergone pairing and before they begin to separate. It occurs through the process of breakage and reunion of your chromatids. And during Pachytene, each chromosome of bivalent has two chromatids, so that each bivalent has four chromatids or strands. So I will show you all these things in diagram soon. So just this is the part of subjective part initially. So generally one chromatid from each of the two homologous of bivalent is involved in the crossing over, and it is assumed that breaking occur at precisely homologous points in the two non-sister chromatids. So here you can see this is bivalent chromosome, homologous one, they are synapsed in this case. So now we have sister chromatids, both of different gene factors, everything in them, and these are all together called tetrads. And in the center they have kinetochores, and they make a tetrad formation. Now during the crossing over, these tetrads could cross like this. Crossing over is happening with the non-sister chromatids, and then these are the non-crossovers chromatids, and then you have exchange of chromatids. So crossover chromatids have exchange of chromatids like this. Understood? So obviously during the crossing over produces two recombinant chromatids called crossover chromatids, or two original chromatids referred to as non-crossover chromatids. The crossover chromatids will have new combination of the linked genes and will be called recombinant. Gametes carrying them will produce a recombinant phenotypes in test cross, which are called crossover types. So frequency of crossing over is equal to number of recombinant progeny from a test cross divided by total number of progeny multiplied by 100. That is called as your frequency of crossing over. So there are different types of crossing over depending upon the number of chiasmata involved. So it is of three types: multiple crossing over, double crossing over, multiple crossing over. So single crossing over, it is refers to the formation of single chiasma between the non-sister chromatids of homologous chromosomes. It involves two linked genes, that is two point test crosses in that case. There's a double crossing over. It refers to the formation of two chiasmata in that case between non-sister chromatids of homologous chromosomes. So it involves three linked genes. Then there is a multiple crossing over, which is occurrence of more than two crossing overs between non-sister chromatids of homologous chromosome is known as multiple crossing over. So, but frequency of such type of crossing over is extremely low. That doesn't happen in high amount. So this is single crossing over. Here we can see there's exchange of material between these two chromosomes like this, blue and red, like that. So second one is the reciprocal double crossing. So you have, they crossed each other, but they have reciprocal cross. So you have a red, blue, red, blue, red, blue, like this. So they are they are cut it out from three different parts have been exchanged: A, B, C, A, B, C. In the earlier case, it was just A was exchanged and B, C was exchanged. Then there's a complementary double crossover in this case. In the second, so they exchange of material, so there's already exchange between these two, A, B, C, like this. And then in the A, it's not in the upper part, the red ones. So you have A, B, C in the blue, and the blue in the blue, A in the blue, B in the red, and C in the blue. But also A and small b and small c to be red and blue, blue. So like this exchange. So here we have double crossing over, in which you have already crossover between first two and first two here, and then you have a resultant like this in that case. So the frequency of crossing over between the linked genes is affected by the several factors: distance between the genes, the frequency of crossing over, so how much they are being frequently crossed over between the two genes is positively associated with the distance between their location in the chromosome. Then there is sex also matters. The frequency of recombination is markedly influenced by the sex of heterozygotes for linked genes. Um, then age of female, that is the frequency of crossing over shows a progressive decline with the advanced age of your Drosophila females. In Drosophila, the lowest temperature also affect. Lowest frequency of crossing over is observed when the female culture at 220 degrees Celsius. If the frequency of recombination increases both at lower and higher temperature than 220 degrees Celsius. So this tends to be increased if we increase the temperature or lower the temperature. Nutrition also matters in the case of crossing over. In the case of Drosophila, which is affected by presence of metallic ions in its foods. Higher the amount, lower will be the crossing over frequency, and vice versa. Chemicals: treatment of Drosophila females with a certain antibiotics like mitomycin D and acetonomycin D, and certain alkylating agent like ethyl methane sulfonate promotes crossing over. Radiation also, an increase in frequency of crossing over observed when the Drosophila females are irradiated with the X-rays and gamma rays. And further, plasma genes. In some species, plasma genes reduce the frequency of crossing over. Example, Tifton male sterile cytoplasm reduces the frequency of crossing in the Bajra. Then there's a genotype, chromosomal aberrations, distance from the centromere. All these are factors affecting your crossing over. What is the significance of crossing over in plant breeding? It increases variability. It helps in breaks the linkages. It makes possible to construct chromosome maps. Then comes your next topic of cytological proof of overcrossing. So in this case, the first cytological evidence in support of genetic crossing over was provided by Kurt Stan in 1931 on the basis of experiment on the Drosophila by using cytological markers. So there is a variety of Drosophila in which part of Y chromosome had broken off and become attached to the end of X chromosome. So usually in Drosophila, normal fly has red round eyes with plus plus, okay, and two mutant genes, one carnation causing darkish red eyes, so CAR, this causes dark eye, being recessive to red plus eye, so they are recessive to red plus eye color, and other bar B causing narrow eyes and dominated with the round plus eyes are both in the X chromosome. And female fly is XX and male flies has XY chromosome. In the female, one X chromosome has been broken into two by X-ray and other by means contain mutant genes called CAR and B. So it has both CAR gene, which is leading to the darkish red eye, and it also has B gene, other bar B, which is causing narrowing of the eyes. And the other X contains normal allele. Here the normal allele is there. So the other X contains normal is for red and for round eye. That is a normal. And its at end segment of Y chromosome was attached. So this Y segment, this one, has been attached to it. This breaking of X chromosome attaching Y segment is a plan or scheme to distinguish between crossing overs and non-crossing over progeny. So this will tell us when we cross with the female whether this crossing of your addition of this and CAR gene and B gene, how they will cross over. So we will discuss about this. So first let's see, we have these non-crossovers alleles. There's a no crossing over happen here, and there is a single crossover. So we made a crossover between these two genes XX. So you have from this normal part plus and B and CAR and plus, so like this. So these are your female gametes, and this is this male has one CAR, then plus, and then normal, other is normal. So like Y gene, this is X, this is Y. So now they are crossed like this. So these are the alleles for the same. So what do you see is that these are the all males, sorry, females, and bottoms are all the males progeny. And this is carnation is there, and there is barred eyes, both are present. So card B and card plus for both. But in this is wild type, this is barred only, this is carnation, barred, wild type, card band eye. So these were the Stern experiment to show you cytological crossing over, that the crossing over that is happening is because of X and Y chromosome. That was done in the Drosophila. So what were the results? Carnation barred female produces 4 types of gametes, out of which 2 are crossovers and 2 non-crossovers, formed with the interexchange of homologous segments of chromosome. When these were mated with the male's XY carnation round eyes, the non-crossing overs carnation and red, while crossing over contains red bar and carnation round. So there is some terms in this crossing overs in the case of plant breeding. The first term is coincidence. It is referred to the occurrence of 2 or more distinct crossing overs at the same time in the same region of pair of homologous chromosomes, and as a result, double crossing over product is obtained. Coefficient of coincidence is estimated by observed frequency of double crossing over divided by expected frequency of double crossing over. So that ratio is called as coefficient of coincidence. And how to calculate coefficient of interference? You just subtract 1 minus coefficient of coincidence. You can calculate the coefficient of interference, which is a crossing over in one region of a chromosome interferes with the occurrence in the neighboring segment. This is known as interference, and the term was coined by Muller. So this is the difference between crossing over and linkage. Linkage we will discuss just now. So there is some differences written there, that crossing over it leads to separation of the linked genes, and linkage it keeps genes together. So both terms looks quite similar, but they have a lot of difference. In crossing over, it involves exchange of segments between non-sister chromatids of homologous chromosomes, whereas in the linkage it involves individual chromosomes. In crossing over, the frequency of crossing over can never exceed its 50%, whereas the linkage number of linkage groups can never be more than haploid chromosome number. Further, it increases variability by forming the new gene combinations. Another case in the linkage, it reduces variability. In crossing over, it provides equal frequency of parental and recombinant types in test progeny, whereas in the linkage, it produces higher frequency of parental types than the recombinant types in test cross progeny. So, chromosome maps can be prepared by genetical or cytological methods. So, genetical method is the general method based upon the crossing over data. Uh, A rule widely followed in plotting genes is that gene A and B are known to be linked. If particular gene is found to be experimental to be linked with gene A, it must also be linked with gene B. So, like this, you can create genetic mapping of chromosomes on the following assumptions: genes, how the genes are arranged in linear order. Crossing over is due to the breaks in the chromatids. Crossing over occurs by chance at random, and the percentage of crossing over between the genes in an index of the distance part. So map distance looks like this. Recombination frequencies between the linked genes are determined from the appropriate test cross. These percent frequencies are used as map units for preparing linkage maps. A map unit is the distance in the chromosome which permits 1% recombinations between two linked genes. A map unit is also called century Morgan, after the name of scientist Morgan, who discovered these linkage maps in Drosophila. So the 5% crossover between gene and B is situated with the 5 map units of distance of the same chromosome. If the third gene C with 7% crossing over is there between A and C, is included in the relationship between, like in the three genes A, B, C as indicated as below. 5 centimorgan, 7 centimorgan, so total between B and C, the crossing over percentage is 12 centimorgan. So like this, you can arrange that. This could also be said like this: A and B, the distance is 5 centimeter, and then B and C, the distance is 2 centimeter. Yeah, and the distance between then A and C will be 7 centimeter. So this we can also say like this to this thing. So we have to choose correct one between these two alternatives. Which one is more informative? The order of arrangement of the three genes and crossing over values between B and C is required. That is quite important. So by cytological studies of chromosomal aberrations and by their behavior in genetical experiments, it is possible to construct map of chromosomes showing the actual physical location of gene in chromosome. Such maps are called as cytological maps of chromosomes. Importance of linkage and chromosome maps in plant breeding. They give an idea whether a particular genes are linked or segregate independently. So linkage intensities can be known and the probability of obtaining a given a combination gene can be assessed. It could help geneticists to play the large and experimental population should be to obtain the plants with desired gene combination. If an easily identifiable qualitative character is found to be linked with the qualitative character can be used to easily identify recombinants. Linkage limits the variability among the individuals. So that was about chromosome maps. We have studied cytological crossing over. We have studied factors affecting crossing overs, mechanism of crossing over. We have studied what is crossing over is. So we have done your this part totally now. Now we are coming to the karyotype and ideogram. Afterwards we come to your CIB techniques. So what is a karyotype? It's a characteristics of chromosomes, complement of eukaryotic species. The preparation and study of karyotype is a part of cytogenetics. The basic number of chromosome in somatic cell of an individual species is called somatic number and is designated as 2n. Thus in humans 2n is equal to 46. Whereas your germline, the sex cell's chromosome number is n equals to 2, n equals to 23. So in normal diploid organisms, autosomal chromosomes are present in two copies and there may or not be sex chromosomes. And polyploid cells have multiple copies of chromosomes and haploid cells have single copies. The study of whole set of chromosomes is sometimes known as karyology. The chromosomes are depicted in a standard format known as karyogram or ideogram in pairs ordered by size, position of centromere for chromosome of the same size. Karyotypes can be used for many purposes, such as to study chromosomal aberrations, cellular functions, taxonomic relationship, or to gather information about past evolutionary events. So this is your normal chromosomes, like they are arranged. Then we arrange with the help of the software of the equal size of chromosomes, because it's 2n is equal to 46, and we arrange from the increasing size to decreasing order, like this. And at the end, these are your autosomal chromosomes and they are your sex chromosomes. So there are X and Y at the end. So from large, 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 going small, 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 like this. And we arrange the identical ones together. One coming from mother, one coming from father. The study of karyotype is made possible by staining. Usually a suitable dye is applied after cells have been arrested during a cell division by a solution of colchicine. So to see them, we have to do staining, and to double your chromosome, we always use colchicine as a medium. For human, white bloods are quite mostly frequently used because they can easily induce these conditions and into the tissue culture and led to the division of non-dividing cells also. Most species have standard karyotypes. The normal human karyotypes contain 22 pairs of autosomal chromosomes and one pair of sex chromosomes. Normal karyotypes for female contains 2X chromosomes and denoted 46, and males have both X and Y chromosome denoted 46, XY. So if there is any difference in the standard karyotype may lead to the developmental abnormalities. So you can stain these absolute sizes of chromosomes by 24-fold between genera same family, and this feature probably reflect different amounts of DNA duplication. So these are 6 different points. Uh, these were like all things telling you about karyotypes. Just go through them. Then comes the ideogram. Ideograms are the systematic representation of chromosomes. They show relative size of chromosomes and their banding pattern. A banding pattern appears when a tightly coiled chromosome is stained with a specific chemical solution viewed under a microscope. So there are G bands and R bands. While so G bands, the one which are stained, and R bands which refuse to have the staining. And the bands can be described location of a genes, interspread elements on the chromosome. So G bands areas are with the proportional more of A and T base pairs. To compare the systematic ideograms, and rendered chromosomes are closed, colored with the AT and GC bases white. The results are interesting, which leads to have these kind of results. So these are G bands dark, and the light bands are the R bands. So this is the karyotype, ideogram, sorry, chromosome 1, chromosome 2, chromosome 3, 4, 5, like this. It goes till your X and Y. Now comes your CIB technique. This method was developed by Mueller for detection of induced sex-linked recessive lethal mutation in Drosophila in male, and this technique C present paracentric inversion in large part of X chromosome, crossing over an inverted portion, and I is recessive lethal. Female with a lethal gene can survive only heterozygous conditions, but if it is in the male, male will die. B stand for the bar I, which act as a marker, help identification of flies. I and B are inherited together, whereas C does not allow crossing over to occur between them. So I and B will be always inherited together, whereas C will not does not allow crossing over to occur between them. The male with CIB chromosome do not survive because of lethal effect. The important step of these methods are as follows. A cross is made between CIB female and mutagen treated male. In F1, half of the females have normal X chromosomes and will survive. Those carrying CIB chromosomes will die. Among the females, half have CIB chromosomes, half normal chromosomes. From F1, females with CIB chromosomes with the normal chromosome are selected for further crossing. So here you can see there is CIB in the female chromosome, and these are all positive, and there is a normal mutagen on this case, and this is male treated. So you made a cross with the male side and female side. So you have these four different gametes are produced. So in this, if CIB, if C is present, then male dies, as we have known, and CIB is always coming together. Like if we multiply this and this, and in this case is a normal male, normal females. Then again we have a second cross between the CLB and mutated and normal male. Then we have again male dies, but in the fourth case, normal male dies if lethal mutation induced. In this case, if this mutation that is induced is lethal, then that mice will also die. So this shows that with the CIB technique, you can check whether the mutation induced in the mutagens treated X chromosomes, remaining half males will also die, resulting in absence of male progeny in the above cross. So this method was induced by Muller and used for unequivocal demonstration of mutagenic of the X-rays. So you can check this mutation in plants by number of plants having the recessive form of trait governed by the gene divided by the total number of plants in the progeny. So that's it. From this perspective, we have covered our topics in this case, and let's go for linkages. So we are done with our first part, second part, and now comes the third part, that is linkage and estimation of linkage. Any questions, students, so far? Linkage maps, they will come soon. We are studying about linkage now. The query will cover in this case. So student, one thing is I have to tell, I mean, today we are lucky that I am having this workshop with you, but my jack for my battery, oh, it's working. Nothing, forget it, what I said. Just a second, give me a pause. I'm trying to fix my charger because I don't want things to be vanished before if my battery goes off and not able to convey. So Sutton Boveri, as we have discussed, chromosomal theory of inheritance. According to the chromosomal inheritance, it is well established that many genes are located in each chromosome in linear fashion. It may be therefore expected that all genes located in the same chromosome would move to some same pole during cell division. As a consequence, such genes will fail to show independent segregation and would tend to inherit it together. And this tendency of genes to remain together in their original combinations during inheritance is called linkage. So Mendel's law of independent assortment is applicable only when the genes are located in different chromosomes, or while linkage refers to these genes located in the same chromosomes. The phenomena of linkage was first reported by Betson and Punnett in 1906. Linkage between genes was first discovered in the experimental with the sweet pea, and they studied flower color, pollen shape in sweet pea involving two varieties races. So if the two races experiments, one parent has purple color with long pollen grains, the other parent has red flower with round pollen grains. So character P, denoted by capital P, is a simple monogenic dominant to the red, while long pollen is dominated to the round pollen with the L. So when these two plants are crossed over, F1 generation would be PL, and PL was purple flowered with long pollen. But in F2, the ratio of four types of plant deviated to normal dihybrid ratio of 9 is to 3 is to 3 1 on the principle of independent assortment of flower color and pollen shape. So here there is a cross, purple and round. So PL, 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 PL. We have purple long again. Then in F2 generation, purple long, purple round, purple red long, red round. So actual numbers coming out like this. Expected numbers that we have done, you remember chi-square method. We calculate the expected numbers, and then expected ratios seems out to be 9 by 16, 3 by 16, 3 by 16, 1 by 16. That is the phenotypic ratio. So the plants with the character combination as in the original parents are known as parental combinations. Plant with the purple flower and long pollen, plant with the red flower and round pollen. Once the crossing over is done, so one character from one parent and another character from second parents are known as recombinant types, or combinations of new combinations of crossing overs. So plants with red flower and a long pollen, that is, then plants with the purple flower and round pollen. So there's a crossing over, new combination is happening, and this is where linkage is playing work. So the chief peculiarity of these results in above table was the parental forms in far excess of the expected number, while their combinations were fewer. This deviation from the expected ratio is due to linkage. So that means purple flower, long pollen are linked because both genes are located on the same chromosome. So your purple flower and long pollen, they were present on the same pollen. Similarly, red flower and round pollen are linked together because genes are located on the same chromosome, and their combination are obtained due to the crossing over between the two concentrated genes in some spore of mother cells. So because they were present on the same chromosomes, so we were so so capital P and capital L. This is small p, small l. So that's why the crossing over, when we do, we saw this 9 is to 3 is to 3 ratio. So this is another experiment. This is now purple round, red long, and when we made a crossover, so here again the parental cross was 9 is to 3 is to 3 is 1, and this division was also due to the linkage, but here our expected numbers were quite less. Yeah, and red round actual numbers were also quite less. So that's why this all is happening because of linkage. Now comes new after the phases of linkage, now comes a new term, that is coupling and repulsion. In the above two examples, it can be seen that one cross, the two dominant factors are linked in one parent and two recessive factors are linked in other. Linkage in such crosses is said to be in coupling phases. While in the second cross, dominant allele of one character pair and the recessive allele of another character pair are linked together in one parent, and the second parent, recessive and dominant allele are linked. Linkage in such crosses is said to be a repulsion phase. So Morgan put forth the theory of linkage and concluded coupling and repulsion were two phases. So types of linkages based on three classified based three types based on crossing over, genes involved, chromosomes involved. So based on the crossing over, linkage may be classified into complete linkage, incomplete linkage. So in complete linkage, it is known that the case of males of Drosophila and the females of silkworms, where there is a complete absence of recombination types due to the absence of crossing over, and in the incomplete partial linkage, if some frequency of crossing over also occurs between the linked genes, it is known as incomplete or partial linkage. Recombinant types are also observed besides parental combinations in the test cross progeny. Incomplete linkage has been observed in maize, pea, Drosophila female, and other several organisms. Then, based on the genes involved, and depending on that, there could be coupling phase, repulsion phase. In coupling phase, all the dominant alleles are present on the same chromosomes, or all the recessive are present on the same chromosomes. So that is your coupling phase. So TR, they are present on your either these are present or these are present. But in the repulsion case, dominant alleles of some genes are linked with recessive alleles on the same chromosome. So like TR. So these are repulsion phase and this is coupling phase. Then based on your chromosomes involved, it could be autosomal linkage or X chromosomal linkage, that is also called as allosomal linkage, sex linkage. So autosomal linkage includes, it refers to the linkage of those genes which are located in the autosomes, and X chromosomal linkage is the linkage which genes are located in the sex chromosomes, that is either X or Y. Linkage involves two or more genes which are located in the same chromosomes in a linear fashion. Linkage reduces variability. So these are various characteristics of the linkages. Linkages may involve either dominant or recessive alleles, or same dominant recessive alleles, or some recessive alleles. It may involve all desirable traits or undesirable traits that we're looking for. It is observed for oligogenic traits as well as polygenic traits. Linkage usually involves those genes which are located close to each other, and the strength of linkage depends upon the distance between your linked genes. That's it. So there's some linkage and pleiotropy. There's a close association between them. That is, close association between the two or more characters may result either due to linkage or pleiotropy or both. Karyotype refers to the control of two or more characters by a single gene. A tightly linkage between two loci can be often confused with pleiotropy. Only way to distinguish between linkage and pleiotropy is to find out crossover products between linked characters. So there are linkage groups refers to the group of genes which are present on one chromosome. In other words, all these genes which are located in the chromosome constitute one linkage group. The number of linkage group is limited in each individual. The maximum number of linkage group is equal to the haploid chromosome number of an organism. For example, there are 10 linkage groups in corn, 7 in garden peas, 7 in barley, 4 in Drosophila, 23 in the men. So these are the various linkage groups. So test cross is the most common method of detection the linkage. In this method, F heterozygotes of two loci is crossed to double recessive parents, and the phenotypic ratio of test cross progeny is examined. If the phenotypic ratio of the test cross progeny shows 1:1:1 ratio, it indicates absence of linkage. If the frequency of parental type and recombinant type deviate from the normal dihybrid test cross ratio, it reveals presence of linkage between two genes under the study. This is now comes the significance of linkage. Linkage limits the variability among the individuals. Linkage between two or more loci controlling different desirable character is advantageous for plant breeders. Linkage is undesirable when desirable and undesirable genes are linked together. The estimates of genetic variances for the qualitative characters are greatly influenced by the presence of linkage also. So we are done with the linkage part also now. So the third part is also done. Now comes your numerical chromosomal aberrations and evolutions that has been done in various species of cotton, wheat, tobacco, triticale, brassica. And we discussed a little bit about structural chromosomal aberrations also. I think this will be better for us for today. This one. Student, I will take a 5 minutes break here. So somehow 5 to 7 minutes. So we see us at 50, 2050 then. Then we start again with this new topic. So student, I'm back. So let's continue with principle of genetics with the chapter numerical mutations and structural chromosomal aberrations. So that is the last topic of your unit 2. Then afterwards we are done for today and we are done for the workshop then. I have to say this with very heavy hearted because it's been like 10 days. Some days I was not available, but we still managed to do so. We've been like a family and going in like this. So let's come to the topic and start with the mutations which alter the chromosome structure, size, or gene arrangement of chromosomal mutations or chromosomal mutations. So chromosomal mutations are widely called as chromosomal aberrations. So there could be change in number of chromosomes, that is euploidy, that is variation in the number of set of chromosomes. One set of chromosome, that is haploidy, that is monoploidy. Then is the polyploidy, three or more set of chromosomes. Then triploidy, tetraploidy, pentaploidy, hexaploidy. So as much they are increasing, septaploidy 7n, octaploidy 8n. It keeps on increasing. Whereas your aneuploidy is the variation in the number of chromosome of sets, that is reduction in the normal number of chromosomes. Monosomic loss of one chromosome, uh, that is 2n minus 1. Double monosomic, that is loss of two different chromosomes, 2n minus 1 minus 1. Then loss of pair of homologous chromosomes, 2n minus 2. An increase in the number of chromosomes, then it's called as trisomy, presence of extra chromosome, 2n plus 1. Double trisomy, two different extra chromosome, 2n plus 1 plus 1. Tetrasomy, an extra pair of homologous chromosome, 2n plus 2. Pentasomy, hexasomy, sapiosomic, like this. The term euploidy designates genomes containing whole sets of chromosomes. Euploids are those organisms which contain balanced set or sets of chromosomes or genomes in any number in their body cells. Euploid is of following types. Chromosome number of chromosome in basic set is called monoploid number. An organism with multiple of monoploid number of chromosomes are called euploids. And eukaryotes normally carry either one chromosome sets or two sets. So humans are diploid creatures, meaning for every chromosomes in our body, there is another one to match it. Haploid creatures have one of each chromosomes. Diploid creatures have two of each chromosomes. Triploid creatures have three of each chromosomes, and polyploid creatures have three or of more each of chromosomes. And it's mostly happening at the non-disjunction level, leading to the production of a lot of more chromosomes, which will help to increase the size, vigor, and increase resistance to diseases. So wheat has 42 chromosomes, but careful study reveals that it is a hexaploid with 6 rather similar but not identical set of 7 chromosomes. Okay, hence 6x is equal to 42, x is 7 in that case. And wheat contains 21 chromosomes, n is 21, and 2n equals to 42 then. Whereas in monoploidy, the monoploid organisms have one genome in other body. When monoploids occur in gametes, it's termed as haploidy. So most microorganisms, they are mostly like these: Datura, Heritium, Sorghum, Triticum. They are all monoploids. And monoploids are usually smaller, less vigorous than their diploid prototypes. Characteristically, monoploid plants are sterile. A reason of sterility is that the chromosomes have no regular pairing partners. For instance, haploid in maize will have 10 chromosomes, and the number of chromosomes in a gamete can range from 0 to 10. Consequently, considerable sterility will be found in monoploid maize. Monoploids play an important role in modern approaches to plant breeding, and they have different loci and they are broken up during the meiosis also. So this is generation of steps showing you how we take anther from a plant and we do a meiotic product cell plated on that, then haploid embryos will grow on that, then haploid plantlets are put up in the agar medium, then you have a monoploid plant at the end. But with the help of a colchicine, and this is an alkaloid drug extracted from the Ottoman crocus, inhibits the formation of mitotic spindles, so cells where the true chromosome sets are produced, and these cells may proliferate to form sector or diploid tissue that can be identified cytologically. So this is your mitosis is happening in monoploid. N is 3 here. So separation is happening, and in the normal case you have two monoploid cells, but with the colchicine treatment you have one diploid cell, so they will not divide, they will remain inside it. So there's no mitotic spindle formation is happening and they remain in one case, so it makes you double the number of chromosomes in that case. So you can use with the plant engineering to produce diploid plants. So you have a sensitive monoploid plant, then you get the somatic cells out of it plated on the selective medium, then you growth on the resistance of plantlet, then leading to the resistance monoploid plant, leading to the repot you add colchicine, and then addition of colchicine leads to the resistance diploid plant, that is fertile plant at the end. Where techniques have been successfully applied to several important crop plants such as soybeans and tobacco, the anther technique for producing monoploids does not work in all organisms or in all genotype of organisms. Another useful technique has been developed in barley, an important crop plant. Diploid barley, Hordeum vulgare, can be fertilized by pollens from diploid wild relative called Hordeum blossom. Then there's a diploidy, which we have two genomes in that case. Diploidy is related with the fertility, balanced growth, great vigorosity, adaptability, and survivability of the diploid organisms. Then comes your polyploids. The organisms with more than two genomes are called polyploids. They have multiple series like 3, 4, 5, 6, known as triploidy, tetraploidy, pentaploidy, respectively. So they are higher than the tetraploids, are commonly encountered in the natural populations. So wheat, strawberries, many commercial plants they include into this polyploids. So examples of polyploids are like oat, coffee, barley, banana, tobacco, sorghum. All these are examples of it. And continuous polyploid series has been reported in rose plant. Basic number is 7. Same way, Charasmathum has basic chromosome number 9 and has diploid 2n is equal to 8, 18. Whereas tetraploids 4n is 36 in that case. Solanum has basic chromosome of 12 and the euploidic series of diploids, triploids, tetraploids are available in different species of it. So how your polyploid is originated? So here you can see first there are chromosomes, then diploid gametes are formed, then haploid gametes are formed, then mixture of diploid and haploid gamete makes a triploid zygote here. So you have three different kind of chromosomes at the end in your zygote, like this. So what things could induce polyploidy? It could be done with the help of physical agents, cell generations, chemical agents. In the physical agents, it could be temperature, centrifugations, X-rays. In the chemical agents, you can use colchicine, like we have discussed, chloral hydrate, acetylphene, veratrine, sulfonyl. Like all these chemicals could be used to increase your number of chromosomes. So autopolyploids, which are composed of multiple sets from within one species, allopolyploids, which are composed of set from different species. So, so these are your fertile allotetraploids, species X, crossed by species Y, AABB. You have diploid. Then, after having diploid sterile hybrid, you add colchicine. This will increase double the amount of your species, and that will be called as fertile allotetraploid in that case. So we can use this technique in various intergeneric species crossovers. So in this case, like roughness and brassica, that is cabbage and radish, which has 2n is equal to 18 and brassica has 2n is equal to 18. So we made a cross with the NN8, NN9 as the gametes, and then sterile F1 hybrid comes out, 2n equals to 18, and fertile amphidiploid 2n plus 2n equals to 18 plus 18. So 4n equals to 36 at the end. So that is also known as Raphanum-brassica, so mixture of Raphanus and brassica, and that is your allotetraploid, which contains 36 chromosomes at the end. Then there's example of Triticum spelta, which has been crossed with the Agalopus squarza. So here you can see that AABBDD made a cross, then you have a triploid hybrid ABD, then with the help of colchins you made a hexaploid of Triticum spelta, like this. So some more example like same in the cotton. So this is a technique showing amphidiploid triticale, like this is rye pollen, which has hybrid first generation and hybrid seed embryos excited. So you made a there's a sprout, then you do a colchicine treatment, chromosomes are doubled, you have a mature plant, then you have a second generation of seeds. There is also called agar culture, root growth, colchicine, chromosome doubling, mature plant, and second generation seeds. So like this you can grow your plants easily. Sorry, you can have amphidiploid triticale like this in that case. And this is an example of alloploidy in the case of brassica. So here we can three different parent species have been hybridized in all possible combination for new amphidiploid species. So here you can see Brassica olenacea species, which is 2n equals to 18, having cabbage, cauliflower, broccoli, kale, kohlrabi, Brussels sprouts, all are included in this. So n is 9 in that case. Then you produced Abyssinian mustard, n equals to 14, and you made a cross with the black mustard of this Brassica nigra. So you made a 2n equals to 34 new species. Also, you produce a leaf mustard with the cross of black mustard and Chinese cabbage, like this. And you made a cross with a turnip, Chinese cabbage cross with this species. You also have new species Brassica napus. So this is how you can do the crosses. And this is another example. You can check it out. So autopolyploids we have discussed how we can do. There are a lot of uses they have. How there are things. Then there's a triploids that is having three. They're part of autopolyploids, arises spontaneously in nature, and constructed genetics by the four extra tetraploids. And triploids, watermelons have been undeveloped seeds. Triploid is also applied in the seedless citrus cultivar. So their triploids are giving us a seedless fruits at the end. So here are there's a showing possibilities of triploids. These are the various trivalent it could happen, or bivalent plus univalent. So these are possible in that case. So autotetraploids that we have discussed, they arise naturally by spontaneous accidental doubling of 2 to 4x. So genome is doubled with the presence of colchicine. So there are large number of chromosomes are present with increased size. So cell size, fruit size, flower size, meta size, all could be large in the polyploidy. So they all are increased in the polyploidy. So here you can see with the help of colchicine 2n, 4n, 8n how the cell size is increasing. The stoma size is increasing in that case. A is your diploid, B is your tetraploid, C is your octaploid. So there's also aneuploids in which chromosome mutations are happening, in which chromosome numbers are abnormal. So there's a part of chromosome set we have in that case. So either they will be increased or decreased, like monosomic or trisomic, nullisomic, all these. So monosomic chromosome complements are generally deleterious for two main reasons. Missing chromosomes perturbs the overall gene balance in the chromosome set. Non-disjunction in the mitosis or meiosis is the cause of the double. Most aneuploids, disjunction is the normal separation of homologous chromosomes. Non-distinction is a failure of this disjoining process and two chromosomes to one pole to learn to the other. Monosomic shows the deleterious effects of genome imbalance as well as the unexpected expression of recessive alleles carried on the monosomic chromosome. So here you can see the non-distinction in the meiosis and mitosis. So in the meiosis, it does not go to other pair; it just remain in the other one side. So meiosis 2 is happening, then they got separated, fertilization happens. So there's a non-distinction, like they are separated in a different way. There's a trisomic is happening. That is, three chromosomes are present, 2n plus 1, and in one case 2n plus 1, 2n minus 1. So that's how trisomic and monosomic things are happening. So this is like as we discussed, n plus 1, n plus 1, there's n minus 1, n minus 1, monosomic and trisomic. So in trisomy there is a 2n plus 1, which is happening because of autosomal, and it's also caused Mongolism in humans also. In plant, first case of trisomy was investigated in the Datura stromonium in somatic cells. In the individuals, they discovered 25 chromosomes in that case. The size and shape and spine characteristics of seed capsules of these trisomic plants have been different seed capsules. Then there's a tetrasomy, double trisomy. These all things are also possible. Now comes the last part, that is structural chromosomal aberrations. So they are cause abnormalities in the chromosomal structures. They alter the structure of the kind of genes present in the chromosomes. These are further classified into four groups based upon the whether they alter the gene sequence, number, or locations. So loss or addition of segments of chromosomes. So deletion, that is loss of segment of a chromosome. Duplication, repetition of segment of a chromosome. Then there's a translocation, inversion. In translocation, exchange of segments between two non-homologous chromosomes resulting in new chromosome. Inversion is a change in the linear order of genes by rotation of a section of chromosome through 180 degrees Celsius. And there could be point mutation, that could be deletion, insertion, substitution, and inversions. So these are the four different kinds of. In the deletion of genes like this, D, E, F, G, they could be deleted and you have remain is A, B, C, H, I only. In the duplication, you have C and D. C and D is duplicated again and added into your chromosome. There could be also inversion. They just change their position invertly by F, E, and D like that. Translocation of genes. This is where information from one or two homologous chromosomes breaks and binds to the other. Usually this sort of mutation is lethal. So like this, J and K, they got attached to it, and from it is removed from that side. And this could lead to the death of the person. There's also mutation and alterations, mutations happening. Insertion is addition of it, and deletion of your chromosome. Frame shift is like you remove first word like the fat cat sat. Then if you remove the T, if you have genetic codes, then half ATC adds that. So it doesn't make sense. There's a frame shift mutation is happening. So that's it for today. We done with the today's course, and I have sent you already all presentations, I guess. It's with you, and anything else, uh, any questions so far from the workshop? Or so students, please follow my subscribe to my YouTube channel also. We will bring more these kinds of workshops, so you can watch them, have a concrete behavior. Like I can bring some more things that you wanted to hear from us. So like, subscribe YouTube channel also. It will be really helpful for me to initiate this small YouTube channel. And thank you very much for being there. I'm humbly happy to be part of your trainer, and to teach you something what I have taught earlier at the university level. So that was the whole course that has been done for you. um, so yeah. Um, Anything else, students? So I think, yeah, we will miss you also, students. Sagar, you were always first person to be say good evening, sir. Actually, I remember that. Thank you, thank you. So soon, maybe in 3 or 4 days, you will get your certificates, and please send your review articles. Only then you will get your certificates and letter for internship also. Yes, yes, please. I want to show you. So we are having more workshops coming. So one is MBBT workshop is coming soon. So molecular biology technique from 7th of September. Ask Shivani ma'am and Vatsal sir about that. If you want to join MBBT molecular biology and biochemistry technique workshop, then there is a workshop of yeah, this one. Sorry, this one is coming from 7th October to 16th October. Then there's a workshop of plant biotechnology is also coming. Biochemistry workshop with and then this one is quite special. This one is proteomics, biochemistry, and bioinformatics. So I will be teaching proteomics and biochemistry in these cases, and please think about these workshops also. So proteomics is there, biochemistry is there, plant biotechnology, molecular biology and biochemistry techniques, MBBT. So these are the various techniques, various are coming us soon. So maybe I can share it here. If anyone is interested, they can look for it later. Oops. Documents, isn't it? Or it's a photo. These are pictures. Think about it. If you want to join us, I will miss you all. I will miss you all. It's become a bond like a family. Before we sleep, we talk with each other. We have enriching lectures about genetics, and then we sleep very smoothly, having these lectures with us. So you can contact me if you have any questions. I will try my best to answer them. If if I'm able to reply, that means I know the answer. If I'm not able to reply you, that means I'm not aware of your things. So you can email me anytime with your questions, and I will try my best to answer your questions, any queries you have about career or anything else. Uh, I will try my best. Okay, with heavy heart, I have to click on end stream now. Um, so, students, thank you so much. Take care and wish you all the best for your future endeavors. Make the best out of it and do the best in your life and keep smiling. Bye bye. Take care. Good night. And I will miss you all. I am emotional fool, so I have to leave now. I might even get emotional. Okay, then bye bye.